Hi, this is Anandhu. I am doing a video abstract on evaluating bona fide pathogenic SNPs using an integrated system genomics and agnostic framework. We know the basics of genetic sequence, predict the structure, structure predict the function. So any kind of changes in the genetic sequence will lead to the next generation also. These kind of mutations we call as heritable mutations. So when we study about mutation, the sequence homology, structural homology, functional or comparative genomics are one of the major areas. How the mutations happens? The mutation happen because of environmental factors like food, exposure to carcinogenic agents, etc. These will lead to epigenetic changes like DNA methylation, histone modification, and chromatin remodeling, which will alter the gene expression and upregulate and downregulate the cellular functions. So mutations can be identified using different NGS methods like whole exome sequencing, whole genome sequencing, and other sequencing methods. So there are 100k plus genome sequence so far. Identifying an associated or pathogenic SNP is one of the major concerns. SNPs or mutations were categorized into different based on the ACMG and AMP guidelines like pathogenic, likely pathogenic, variant of unknown significance, likely benign and benign. Over the last decades, the databases like ClinVar, CARD, GURP, etc. were used to identify the pathogenic mutations which were stored in VCF files. Search in these databases in a local instance need a rigorous downstream validation which is heavily dependent on the database, which means is a dependent framework. For processing and analyzing the NGS data, we use different pipelines and platforms. So our objective to develop a web resource with agnostic framework for ascertaining candidate mutation from a vivid NGS data, and to validate the agnostic or prioritized framework with a various disease phenotype using machine learning heuristics. Although there are databases and web servers are there to screen pathogenic SNPs, most of them are dependent framework. Therefore, we would like to fill the gap by developing a web server with an agnostic framework. So we use different types of data sets like public, whole exome sequencing and NGS data, and our own lab data and another 100 databases, and we do that downstream validation. So this is one of the our proposed framework after the NGS data processing and analysis, identifying the concerns and filtering out the false positives, and train our in-house made model and generate an agnostic framework. The conclusion and future of our study is to develop a comprehensive marker catalog specific to mutation from end user based on the mutation, develop a standard operating procedure for validating mutation, a web server with an agnostic framework for ascertaining candidate mutation from NGS data. So thank you. We look forward for your questions.